Good morning to my respected teachers, seniors, and colleagues. My topic for today's seminar is pharmacogenetics. In this seminar, we will see introduction, definition, genomic basis, genetic variation, which includes single nucleotide polymorphism, insertion deletion, then ethnic diversity, pharmacogenetic phenotypes, tools for gene tracking in DNA, and role in clinical practice. Introduction. There is a great person-to-person variation in the response to drug therapy, and to predict the effectiveness or safety of a drug is very difficult. Interpatient variability in drug response may be due to genetically determined differences in drug metabolism, drug distribution, and drug target proteins. The influence of genetics on drug response was demonstrated in 1956 with the discovery of inherited deficiency of G6PD. It was responsible for hemolytic reactions to the primaquine. Pharmacogenetics is defined as the study of the genetic basis for variation in drug response. The term pharmacogenetics is often used interchangeably with the pharmacogenomics, but there is a difference. Pharmacogenetics is study of monogenetic variants that affect drug response, whereas pharmacogenomics is the study of the entire spectrum of genes that interact to determine drug efficacy and safety. We will take carvedilol as an example. A pharmacogenetic study would examine the influence of the beta-1 adrenergic receptor gene on blood pressure response to carvedilol, whereas a pharmacogenomic study will examine the interaction between cytochrome P2D6 and beta-1, beta-2, and alpha-1 adrenergic receptor genes on carvedilol effects. Multiple proteins and genes are involved in determining the ultimate response to most drugs. The goals of pharmacogenetics are to optimize drug therapy and to limit drug toxicity based on an individual's genetic profile. Pharmacogenetics aims to use genetic information to choose a drug, dose of the drug, and the duration for which the treatment is to be given. This will have the greatest likelihood of achieving therapeutic outcomes with the least potential for harm in a given patient. Important genetic concepts. Now, in the formation of the DNA, there are two purine nucleotide bases, adenine and guanine, and two pyrimidine nucleotide bases, cytosine and thymidine. Three consecutive nucleotides form a codon. Gene is a series of codons that specifies a particular protein. Allele is a sequence of nucleic acid bases at a given gene chromosomal locus. In most cases, an individual carries two alleles, one from each parent, at each gene locus. Two identical alleles make up a homozygous genotype, whereas two different alleles make up a heterozygous genotype. Phenotype. It is an observable characteristic or physical manifestation of the genotype. Types of genetic variation. Polymorphism. It is a variation in the DNA sequence that is present in the frequency of 1% or greater in human population. In contrast, if the variation is less than 1% of the population, it is known as mutation. It causes inherited diseases like cystic fibrosis, hemophilia, and Huntington disease. Common diseases such as essential hypertension and diabetes mellitus are polygenic. These are due to interaction between multiple genetic polymorphism with environmental factors responsible for disease susceptibility. Types of genetic variants. Variation in human genotype are of two types, SNP and indels. SNP, single nucleotide polymorphism, is single base pair substitution in greater than 1% population. SNPs occur when one nucleotide base pair replaces another. Thus, SNPs are single base differences that exist between individuals. Indels, insertions or deletions. Indels can be short repeats in promoter or insertion or deletion that add or subtract amino acid in the coding region. Single nucleotide polymorphisms are of two types, coding region SNP and non-coding region SNP. Coding region SNP is further divided into non-synonymous, synonymous, and nonsense. If the base pair change results in an amino acid substitution, it is called as non-synonymous or missense. If the base pair substitution within a codon does not alter the encoded amino acid, it is called as synonymous or sense. In nonsense, if the base pair substitution leads to a stop codon. There are 64 trinucleotide codon representing 20 amino acids. So multiple codons encode the same amino acid. Often the substitution of third base pair known as vowel position do not alter encoded amino acid. Almost 10% of SNPs display more than two possibility in alleles but no change in phenotype. For example, change in codon CCG to CCA will encode proline amino acid only. Non-coding region polymorphisms may occur in the 3' and 5' untranslated regions, in promoter or enhancer regions, in intronic regions, or in intergenic regions. The vast majority of human DNA, more than 97 to 99%, is non-coding. Non-coding SNPs in promoter and enhancer region may alter DNA binding for transcription. 3' SNPs may alter binding of microRNAs, which may affect stability of a transcript. Ethnic diversity. Polymorphisms in human populations can be cosmopolitan polymorphisms or population-specific polymorphisms. First is cosmopolitan polymorphisms. It is present in all ethnic groups found at higher allele frequencies than population-specific polymorphism. The population-specific polymorphisms are likely due to migration of human populations. They are consistent with geographical isolation of human populations. Pharmacogenetic study design. A pharmacogenetic trait is any measurable or discernible trait associated with a drug. For example, enzyme activity, drug level in plasma or urine, blood pressure lowering effect of a drug, or drug-induced gene expression patterns. Most of the pharmacogenetical traits are multigenic. Therefore, considerable effort is being made to identify the important genes and their polymorphisms that influence variability in drug response. Most genotypic methods use germline DNA, that is DNA extracted from any somatic diploid cells, usually WBCs or buccal cells. Genotyping tests are directed at each specific non-polymorphic site using strategies that generally depend at level on the specific and avid annealing of one oligonucleotide to a region of DNA overlapping the polymorphic site. The tools of gene tracking, analysis of DNA, restriction enzymes. These enzymes cut DNA into small pieces. DNA probes. Probes are single-stranded specific sequences of DNA. Hybridization method. It is a technique that measures the degree of genetic similarity between pools of DNA sequences using probe DNA. PCR, polymerase chain reaction. In this technique, a fragment of DNA is isolated and with the use of enzymatic replication, it is amplified exponentially. Southern blotting. It is a technique for separation of DNA fragments by electrophoresis and then identification of a target fragment with a DNA probe. Because genomic variability is so common, cryptic or unrecognized polymorphisms may interfere with oligonucleotide annealing, and this results in false positive or negative genotype assignments. Therefore, we require high standards for genotyping technology, perhaps with more than one method required for each polymorphic site. Candidate gene approach versus genome-wide approach. Candidate gene approach. This study aims at identifying the genetic polymorphisms that are likely to contribute to the therapeutic or adverse responses to the drug. After genes in the drug response pathways are identified, there are some databases that contain information on polymorphisms and mutations in human genes. For example, PharmGKB, Pharmacogenomics Knowledge Base, is a genotype and phenotype data related to drug response. DBSNP is maintained by NCBI and is for SNPs and frequencies of SNPs. HGV base Human Genome Variation Database is for SNPs and polymorphism. A potential drawback of this approach is that the wrong genes may be studied. In candidate gene association study, there are two categories of polymorphisms. First one, the polymorphisms that do not alter the function of expressed protein. Rather, these polymorphisms are linked to the variant allele that produces the altered function. The other one is causative polymorphism, which directly precipitates the phenotype. For example, a causative SNP may change an amino acid residue that may result in a protein that is non-functional or has reduced function. Genome-wide approaches provide unbiased survey of the genome to identify previously unrecognized candidate genes. For example, RNA, DNA, or proteins from patients who have unacceptable toxicity from a drug can be compared with RNA, DNA, or proteins from identically treated patients who did not have such toxicity. Pharmacogenetic phenotypes. Candidate genes for therapeutic and adverse response can be divided into three categories. First is pharmacokinetic genes which modify drug disposition. The second is receptor or target, those who alter the function of the molecules with which drugs interact to produce their beneficial or adverse effects. And the third one is disease modifying. Disease modifying are those altering the broad biological milieu in which the drugs interact with target molecules, including changes associated with the disease for which the drug is being prescribed. Pharmacokinetics. Germline variability in genes that encode determinants of the pharmacokinetics of a drug concentration, and are therefore major determinants of therapeutic and adverse drug response. For example, cytochrome P2D6 is involved in metabolism of several tricyclic antidepressants. There are 7 variant alleles. Those are account for more than 90% of the poor metabolizers. They are low activity alleles. Therefore, the drug is metabolized poorly, and therefore the response of the drug will be exaggerated. There are 13 variants alleles account for rapid metabolizers. Those are high activity alleles. Therefore, the drug will be metabolized rapidly and it results in diminished drug response. Cytochrome P2C19 is involved in metabolism of proton pump inhibitors. Few single nucleotide polymorphisms lead to deficient and poor metabolizer phenotype. This will result in higher exposure to parent drug and greater pharmacodynamic effect. As a result, there will be higher probability of ulcer cure. Cytochrome P2C9 is involved in warfarin metabolism. Inactivating polymorphisms in this lead to low activity variants. The clearance of warfarin will be lower. Therefore, warfarin will remain in the system for a longer duration and at a higher concentration. This results in higher risk of bleeding complications. Enzyme UGT1A1 is involved in metabolism of irinotecan by glucuronidation. A promoter region variant of this enzyme leads to reduced transcription rate of the enzyme. Because of reduced glucuronidation, there will be higher level of the metabolite SN38. Because of higher level of this active metabolite, there will be toxicity of the drug. Enzyme TPMT methylates mercaptopurine. 3 single nucleotide polymorphisms lead to inactivating alleles. This will result in reduced methylation of the drug. Therefore, the drug will remain longer in the system. This will lead to higher risk of toxicity of the drug. Targets and receptors. Gene products that are direct targets for drugs have an important role in pharmacogenetics. For example, rare point mutations in MTHFR cause severe mental retardation and a shortened lifespan. These are some examples of receptors or targets that are gene products with their respective drugs and responses. The third is disease modifying polymorphisms. For example, statins and tacrines interact with apolipoprotein E and will have lipid lowering effect and clinical improvement in Alzheimer's disease. Abacavir will interact with HLA-B 5701 and M493D and will cause hypersensitivity reactions. Oral contraceptive pills will interact with prothrombin factor 5 and will cause venous thrombosis. Pharmacogenetic and drug development. Genome-wide approaches hold promise for identification of new drug targets. For example, by identifying genes whose expression differentiate into inflammatory process, the compounds could be identified that lead to gene expression, and this compound could serve as a starting point for the anti-inflammatory drug development. Pharmacogenetics may identify subset of patients who will have high or low likelihood of responding to an agent. For example, somatic mutation in EGFR gene in lung cancer is likely to respond to tyrosine kinase inhibitors like gafitinib. With the help of pharmacogenetics, we can identify genetic subsets of patients who are at high risk of serious adverse events, and we can avoid testing such patients for that particular drug. For example, identification of HLA subtypes associated with hypersensitivity to abacavir could identify a subset of patients who should receive alternative therapy to abacavir. Pharmacogenetics in clinical practice. The potential utility of pharmacogenetics to optimize drug therapy is great. Once adequate genotype studies are done, molecular diagnostic tests will be developed that detect more than 95% of important variants for majority of polymorphisms. The advantage of tests are that they are needed only be conducted once in the lifetime of an individual. The data collected from pharmacogenetic studies will demonstrate whether dosage individualization can improve outcomes and decrease short or long-term adverse effect. Thank you.